Welcome back, class. We're in the middle of chapter 15. We have talked about how a microbe can get inside of a host, how it can adhere, how it can avoid the immune system, and how it can get inside of a cell and arrange actin. All of this, though, it could do without causing disease. So now we have to start talking about what it's actually doing that makes it pathogenic. Some different things that a bacteria could do to damage your cell is one, it could use the host cell's nutrients. So if you have nutrients that your cell needs to be healthy and the bacteria takes it away, your cells are no longer going to be healthy. One is by causing direct damage wherever it invaded, just killing cells, destroying tissues, wherever it's going. One is by producing toxins. Toxins are probably one of the big topics today. And one of the things about toxins is that they can travel through blood and lymph, so it can damage parts of the body far away from where the bacteria are actually growing. And one is by inducing hypersensitive reactions, which is another way of saying the bacteria itself isn't doing anything bad, but your immune system is overreacting, and that is going to cause harm. One way that a bacteria can use up your nutrients is by producing something called siderophores. So many pathogenic bacteria are going to need a lot of iron to grow, but we don't have a lot of free iron just hanging around in our body. Most of it's bound to carriers like hemoglobin in your body. And so that bacteria is going to find a way to play tug of war to take the iron into the bacteria. And siderophores are a type of iron-binding protein. They can be secreted in the medium, take the iron up from the carrier, and then bring that iron into the bacteria. Sometimes just the iron is taken up. Sometimes the carrier will also be taken up, so iron and hemoglobin. Sometimes a toxin will release the host cell. In the process, the iron is released, and the siderophore is able to bring it into the bacteria. Direct damage could involve a pathogen growing inside of a cell, and when it has grown enough, it is going to cause a cell to rupture. That means the cell is going to just be destroyed. That allows the bacteria to spread to other tissues. Some bacteria will initiate phagocytosis to be able to be taken into a cell, and then will disrupt the cell. Some pathogens will produce waste products. But most damage is going to be done by toxins. Toxins are poisonous substances that certain microbes can produce. They can produce fevers, cardiovascular disturbances, diarrhea, a whole bunch of things you can read here. Toxogenicity is the ability of a microbe to produce toxins. Toxemia, I think we've used that word before, is the presence of toxins in the blood. And intoxication is the presence of a toxin without microbial growth. So intoxication, I think of like someone is making chicken. They haven't washed their hands, but they're cutting up the chicken, and the bacteria from their hands gets onto the chicken. The bacteria start growing, and as they're growing, they are producing toxins. The chicken gets cooked, the bacteria gets killed, so no one is eating any living bacteria, but that toxin is still there. So that's a form of intoxication. Digesting a toxin, having a toxin in the body without having any microbes growing. Toxins can be divided into two groups, exotoxins and endotoxins. Exotoxins are proteins. That is important. They are made of proteins. They are produced inside bacteria. It's just a normal part of their growth and metabolism. So as that bacteria is growing, it's just doing whatever it's doing to grow, and something it makes happens to be toxic to us. Those toxins might be secreted into the medium as a cell is growing, just as a normal part of its growing. They might be released after the cell dies and is lysed. Exotoxins tend to be easily diffused into the blood, and so they can transport through the body easily. And then they can destroy parts of the host cells. They can inhibit metabolism of our host cells and end up causing harm. Many of the diseases that bacteria if a bacteria produces an exotoxin, it's likely that the disease is caused by the exotoxin and not caused by the bacteria themselves. And so sometimes, instead of creating vaccines that target the toxin, instead of creating vaccines that target the bacteria, we create vaccines that target the toxin, because that's the dangerous part of this combination. Antitoxins are antibodies that our body produces that bind to toxins to give us immunity to exotoxins. Toxoids are inactivated toxins that are used as vaccines to stimulate the production of antitoxins to produce immunity. So both the diphtheria vaccine and the tetanus vaccine are toxoid vaccines. That means they are made of inactivated toxins. Because they're inactivated, they cannot cause the disease, but they stimulate the production of antitoxins, those antibodies, so that if you ever are exposed to diphtheria or tetanus, your body will be able to control those exotoxins instead of getting sick. Exotoxins come in 3 general groups: AB toxins, membrane disrupting toxins, and superantigens. AB toxins have 2 parts, an A part and a B part. A stands for active, B stands for binding. So the B part binds to the cell, and then the A part gets inside the cell and causes all kinds of damage. Membrane disrupting toxins will disrupt a membrane. So they will lyse cells, create a channel through the membrane. Some bacteria use this to escape a phagosome, so they are able to avoid being phagocytosed. We'll come back and talk about phagosomes in our next lecture, so sit tight if you're uncomfortable with that term. But it allows bacteria to escape being phagocytosed. And then superantigens are toxins that cause intense immune responses. So they cause our cells to overproduce cytokines and have just a cascading effect in our body. It can lead to fever, nausea, vomiting, diarrhea, and at its worst, shock and death. Here's an example of an AB toxin. Here's a bacteria, a huge bacteria. Usually they're way tinier than our cells, but it produces this toxin, an AB toxin. Here's the A part, the active part, and here's the B part, the binding part. So the B part binds to your cell, gets brought into the cell, and the A part then goes to work. Here, this A part is stopping a protein from being made. So your cell is not making a protein it needs. It's suffering and is a consequence. Here's a list of diseases caused by exotoxins. It's a nice list for you to read through. I encourage you to pause it and read through it to be familiar with these. These are diseases you are likely to encounter as a practitioner, so you should really go ahead and read through these. Then, in contrast to exotoxins, there are endotoxins. Endotoxins are lipids. That's very different from an exotoxin. They are lipids. Specifically, they are lipid A. They are the lipid part of lipopolysaccharides, which is part of the outer membrane of the cell wall of gram-negative bacteria. This should be familiar to you because we did talk about this in chapter 4. We talked about gram-negative cells having an outer membrane. That outer membrane has lipopolysaccharide, which includes lipid A. That should be a review for you. And if it's not, please go back and review that. Because it's part of their cell wall, it is going to be contained on the bacteria as the bacteria are growing. But when the bacteria are dying or undergoing lysis, parts of the cell wall can be released. And because it's a consistent lipid, it's a consistent molecule, they all will produce the same signs and symptoms, including chills, fever, weakness, aches, sometimes shock and death. It can create miscarriages. Not necessarily to the same degree for different infections, but that same set of illnesses. Endotoxins can also go on to create a couple of other really severe issues. One is disseminated intravascular coagulation, which is where the endotoxin will start activating blood clotting proteins. So the patient starts getting blood clots all over their bodies. It shows up as small purple little bruises all around their body. Because their blood clotting proteins are being used in making clots, it decreases the blood supply available to the tissues. Endotoxic shock is a life-threatening decrease in blood pressure that the endotoxins will increase the permeability of the capillaries. So as your capillaries are increasing their permeability, they're more permeable. That means they're leaky. Fluid is going to leak out of those capillaries, so there's not as much fluid left in the veins. Without that fluid, the blood pressure crashes. You can see DIC or endotoxic shock in Salmonella typhi, Proteus, and Neisseria meningitis. Endotoxins can also create a fever. This is called the pyrogenic response. So here's a little bacteria, and here's a macrophage. This macrophage is going to perform phagocytosis, or eating, breaking down that bacteria to prevent an infection. As it starts breaking down the bacteria, pieces of the cell wall, that lipopolysaccharide, are going to be broken down off of that. That is going to cause the release of cytokines. Cytokines are like little text messages that can travel from one cell to another. These cytokines are going to travel through the blood vessel to the brain, to the hypothalamus. The hypothalamus will release prostaglandins, which will result in a fever that raises the body temperature to a higher temperature. A fever is a nice way to help disrupt an infection because it raises that temperature where bacteria can't grow as well, so that it's a little bit easier to break down that infection. Okay, comparing exotoxins and endotoxins, both this slide and this table on the next page are really important. You can expect a lot of questions about exotoxins and endotoxins, not only on your quiz for this chapter, but also in your next exam. Exotoxins are typically proteins produced inside bacteria. Endotoxins are the lipid part of gram-negative bacteria. So where endotoxins are only found in gram-negative, exotoxins are commonly found in gram-positive. They can also be found in gram-negative bacteria, algae, fungi. Anything can create a toxin. So exotoxins found in gram-positive and gram-negative, endotoxins gram-negative only. Exotoxins are just a metabolic part of a cell. Endotoxins are part of the cell wall, the outer cell wall. Exotoxins are proteins. Endotoxins are lipids. Exotoxins are unstable at high heat, so not surprisingly, they do not induce a fever. Endotoxins are heat stable and do produce fevers. Exotoxins are very toxic. That means it takes only a little bit, so the lethal dose is small. Endotoxins have a low toxicity, so the lethal dose is very much larger. Exotoxins can be converted to toxoids to create a vaccine. They can be neutralized by antitoxins. Endotoxins are not easily neutralized, so we don't have effective toxoids. So now you should be able to tell me, what are siderophores doing? Describe, differentiate exotoxins and endotoxins. And food poisoning can be divided into two categories: food infection and food intoxication. On the basis of toxin production by bacteria, explain the difference between these two categories. Okay, I'll come back to you. I'll see you in our next video. We're going to talk a little bit more about how microbes can cause disease. We haven't even started talking about viruses or other pathogens. And then talk about bacteria or how they leave our body. All right, I'll see you later. Let me know if you have any questions. Bye.